All right. Hello, everyone. Let me send a notification to my main channel that I'm live, and then I'll get this discussion going. So give me 1 second here. Let's see. Okay. So we're live. All right. I see some likes already. I see a couple of people in here. Feel free to go ahead and comment if you're in. Let's see. I'm going to share this. For my discussion on epidemiology. All right, that's been shared. Now let me switch channels here. All right. So I will go ahead and start. All right. Hello, everyone, and welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this stream, please share it, and please subscribe to my channel. I saw that I got Tasha's here. Hey, Tasha. I see that I got a bunch of new subscribers today. I think some of those were from the Roger Report. And over on my original channel, I got a bunch of new subscribers after my discussion with BGS on Sunday. So Big Discussions 76 Science and Technology is now at 803 subscribers, which means that I'm 197 away from 1,000. And if I can get to 1,000, that would be huge because, well, not only would I have a super chat for this channel, but I would also have a community tab. So once I have a community tab over here, I can share more information even without going live or doing pre-records. So if you're new and you're watching this live, and if you're watching this on the playback, please definitely hit that subscribe button. Hit the like and hit the subscribe button. That'd be very, very helpful for me. Okay. So today I'm going to have a discussion with a special guest about the field of epidemiology. And so we're going to go for about an hour. And for the folks who come in, if you have questions, you can leave those questions in the chat. I'll address them once I get done with my interview questions. So epidemiology. So why is this all important? So I created this channel for a couple of reasons. Number one, to have science discussions, and number two, to increase awareness of the STEMs for individuals like myself and for individuals individuals with backgrounds like my own. And so I'm one of the few STEM professionals in my family. So when I was growing up, I didn't really know any. So I had an aptitude for biology and those related fields, but there weren't a lot of STEM. There weren't really any STEM professionals in my my home ecosystem. And as I told another peer here in the YouTube streets, it wasn't until I got to graduate school at the University of Michigan, shown in this picture here, it wasn't until I got to Michigan that I even heard of an institution like MIT. So there are whole communities that have never heard of MIT, or I mean, you hear of Georgia Tech because of the basketball team and because of the football team, but you don't really hear about these institutions in certain communities. So that's one of the reasons why I created this channel. And so tonight we're going to talk about epidemiology. But before I bring my guest up and we jump into that, I'm going to just give a little bit of a background so as to set the context and so that we have a good understanding of why this is important. Hello, Corrine Henry. So hello, Tasha. And also hello, Corrine. Thank you for being here. So as many of you know, my background is in pharmacology. And I wrote a post about pharmacology, discussing it at a high level on my blogging platform. This blog post is below in the description box in case you want to read it for yourself. And my training is actually more in the realm of the basic sciences. I see Go Professor's here. Hey, Go Professor, thank you for stopping through and showing some support. So pharmacology. Pharmacology has a clinical side and it has a basic science / research side. So it turns out I also wrote a blog post on the basic sciences. So the basic sciences, when you think about the basic sciences, that blog post is here. When you think about the basic sciences, you want to think about research. You want to think about the pursuit of new knowledge. When it comes to the biomedical sciences, that for the most part takes place in a research lab, doing experiments. For me, it was running gels, expressing protein, doing Western blots and those kinds of things. And I described part one of my graduate thesis a couple of months ago. And my graduate thesis involved this enzyme here, neuronal nitric oxide synthase. And what we were trying to do was we wanted to figure out what this drug, guanabens, did to this enzyme, neuronal nitric oxide synthase, which targeted it in a more pronounced way to the 26S proteasome system. So that was my research project. That was a basic research project in the lab, performing experiments, asking specific scientific questions. And afterwards, I've been cutting off little pieces of that live stream, focusing on some of the techniques, such as the transfection of cells, such as column chromatography, and SF9 overexpression systems and protein purification. But that's all, in the basic research realm, the basic science realm. That's one aspect of exploring human health, figuring out how things work. For me, that involved drugs. So that's pharmacology. And in some instances, you're using an animal model to ask those questions. And in some instances, you're using in vitro techniques. In some instances, you're using cells. So the basic sciences and the basic research disciplines, that's one aspect of human health. Now, there are other aspects of looking at human health and how to protect human health. Another aspect is the regulatory sciences. I'm going to show you one more blog post here. I wrote this because not everyone knows what this is. So the regulatory sciences. For the regulatory sciences, you're usually looking at the generation of a consumer product, and you're trying to figure out the safety of that consumer product. Here you see a picture of boxes of toothpaste, but the product could be a drug. The product could be some other sort of industrial chemical, like a pesticide or jet fuel or a textile. But the point is that you're asking questions about human health, and you're trying to determine the safety of these products before you sell them to humans. So those are the regulatory sciences. And there are regulatory scientists in the private sector and industry, and there are regulatory scientists in the federal government and in state governments. So there are multiple ways to look at human health. Now, it turns out that there's another aspect of looking at human health, which is the field of epidemiology. This is the thumbnail for this live stream, and what you're looking at is this depiction shows a population of people, and you have the gray people, which are the majority, and you have a minority or a subsection of people there, which are the green people. And I think with that cartoon, my guest can probably elaborate on this. I think what that cartoon is trying to tell us is that, well, with my research background and my current career in the regulatory sector, we know that there are genetic variances within the population. We know that there are cultural differences within the population. We know that there are ethnic differences, and we know that there are all these kinds of differences within just the population within the United States. And those differences make us susceptible to drugs, injury through drugs. Some of those differences make us more susceptible to things like cancer. And some of those differences make us more susceptible to things like asthma. So there's another aspect of human health that just looks at susceptible populations and why certain populations are at risk for disease versus other populations. So that's the field. That's my interpretation of the field of epidemiology. In my current regulatory role in the federal government, there are actually epidemiologists in my division. I've worked with them, so I have a bit of a knowledge of what they do, but I thought it would be fun and educational to have someone who was actually trained in the field to come to my channel and actually talk about it in greater detail. So with that, I'm going to bring up my guest. I want to welcome Tamera Travis.
Hello.
Who has training in epidemiology, and she's going to talk to us about her studies in epidemiology and some of the research that she's done, and she's going to talk to us about the field as well. So Tamera, welcome.
Thank you. So yeah, I think that your interpretation of the diagram in the background is very good, and it can also be interpreted as infected people within a population. So the green people could perhaps have an infectious disease, and everybody else, the gray people, are perhaps susceptible. So I think, yeah, that there are a lot of ways to interpret that diagram, and there are a lot of ways to use epidemiology. So I'm excited to talk about it with you.
Okay. So everyone, I'm going to let you in on a little secret. Okay. So me and Tamera, we are relatives. So that's how. Well, she's another scientist in the family, so that's how we know each other. So I think I had my brother on here one time for something, and in any case, that's how we know each other. So we go way back. So Tamera, so thank you for being here. I think this will be a fun and educational discussion, and you've seen the questions I'm going to ask you, and I think you have some things to show us as well. So I'm going to go to, you have a welcome here from Corinne Henry. She says, welcome to Tamera. So the questions I had here. So I'm going to start with the first one. So in as much detail as you want to go into, talk about your educational path and your background and what got you interested in science.
Yeah, so I was very fortunate in my elementary school, middle school, and high school. I was a part of these accelerated programs, and in those programs, we had the ability to go and touch on different types of sciences that weren't normally covered in school curriculum. So we had like an underwater marine biology course and all of these other like strange courses that normal students don't get to do, but I had the privilege of doing. I also grew up in Georgia, and obviously the CDC is in Atlanta. So I got to go to the CDC very often and kind of get that kind of exposure to diseases, outbreak control teams. We got to try on hazmat suits, that whole thing. So I was able to be exposed to a lot of different types of sciences from a very young age, which I'm incredibly grateful for. So that's kind of my immersion into science in general.
Okay. So you got to go to the CDC at an early age.
Mm-hmm. Yeah, so you can do tours, and they've got really great tours there. They've got what do they call them? Outbreak detectives that you can do like for summer camps. There's a disease detective app that you can get for the CDC. So I was up with all of that from very young.
Okay. So just for context, for the audience, I'm from Buffalo. So we don't have the CDC in Buffalo. So that's huge to be able to go to the CDC because you always hear about it in the news,
Yeah.
Particularly with what's going on now. But anytime there's I think years ago we had the swine flu and the avian flu, and you always hear about the CDC. So to be able to go at a young age, that's huge.
Yeah.
Okay. Was there any particular course that you were that was your favorite, or that you were good at, like physics or biology or anything, or were you good at all of them?
I think I was reasonably good at all of them. I'm kind of still in elementary school here, so we didn't really go into like biology and things of that nature. And I wanted to be an epidemiologist when I was like 6 or 7. My mother could attest to this. And that's because there was a TV show called Monsters Inside Me that used to be on Animal Planet. And basically it's these people who have infectious diseases, and it walks through their process of figuring that out. So it'll be, oh, I woke up one day, I had a massive headache, my stomach hurt, blah, blah, blah. I went to the doctor, this is what they said. And the process of it being diagnosed. And that's kind of what got me into epidemiology. I know I'm kind of bleeding into the next question a bit, but that kind of figuring things out, that critical thinking was really interesting to me. And that's kind of what drew me in. And that's what I knew I wanted to do.
And what's the name of the show again?
Monsters Inside Me. You can still find it like on YouTube and stuff, I'm sure.
Inside Me. Okay, I'll have to take a look at that. All right. So from an early age, you wanted to get into epidemiology.
Mm-hmm.
So I know that you had a very exciting and unique educational path, which basically involved you going overseas. Can you talk a little bit about how you did that and what that entailed?
Yeah, of course. So again, wanted to do epidemiology. The only problem is it's very difficult to get undergraduate degrees in epidemiology. I think there are maybe 2, 3 programs like ever that exist with that. So you have to choose a pathway. So I chose public health, and I went to the UK to do it. In your senior year of high school, you get all these pamphlets, all these emails from all these different schools saying that you should apply. And I got one from the University of East London, and I decided to look into it a little bit more. So schools in the UK, undergraduate degrees are 3 years instead of 4, which was really appealing to me. That's a whole year off. Also, you don't have any general education classes like you do in the United States, where you've got like lecture halls of 500, 600 people, and they're like, I don't know how anybody can learn in that environment, but they don't have that in the UK. So you jump in and you do exactly what you want to study, which is really great for me. The class sizes are smaller, and it's also cheaper than the United States. So I just kind of thought, why not, basically, and decided to take the plunge to go to England to study. And I got my degree in public health there. And that was really transformative for me in terms of shifting from just epidemiology to more population health and public health with epidemiology, because epi is just diseases, right? It doesn't always include the wider context of things. And there's this social determinants of health model, which basically means that you've got yourself in the center and factors that you can't change, like your age, your sex, and then it's individual lifestyle factors, and then community networks, and then where you live, where you work, and then the government. All of these things act upon you and determine how or if you will develop a disease and how you'll be able to get treatment for it. And having that wider context was really, really helpful to me in narrowing down what type of epidemiology that I wanted to go to. So after I finished my degree, I went to Peru for a little bit and did a Foundations of Global Health, public health project there for a bit. And then I continued my education.
Okay, so you said, wow, that was a handful.
I know, I'm sorry.
All right, we have a special comment here. Good evening, son and niece. That's my mother, everyone.
Hi, Auntie Wendy.
So my mother edits most of my stuff, so, and I probably got my writing gene from her, so that's my mother. Okay, so you, wow, you said a lot there. So let's go back. So if someone wants training in epi, they would have to go to a school of public health. Yes?
No. Okay, so you don't have to go to public health. There just are not very many epi undergraduate degrees. So I'll touch on this a little bit more when you ask me about what epidemiology is, but there's just kind of building blocks that you can take, and epidemiology is not the first block, if that makes sense. And again, I'll explain this a little bit more later. Okay,
so that means that in the United States, individuals would get their bachelor's in something else and then go get their graduate training.
Typically, yeah.
Okay, and so going back, so you said that for your in your education, in your track, you got training in epi and something else, or it was two different kinds of epidemiology?
Well, there are loads of different types of epidemiology, or rather applications of it. So in my training, it was, so epidemiology is just statistics. So I guess I'll kind of clarify that there, is that it's really calculating risk rates, odds ratios, attack rates. So what is the likelihood that you will get a disease? And you can do that on a micro level, on a population level, and you can apply that in different ways. So you can apply it to a neighborhood, again, an entire population. You can look at specific risk factors. So within public health, that looks at all things that are not you personally. So again, like where you live, where you work, even what government you live under, that kind of thing. Whereas epi can only focus on individual factors. If that makes sense. Feel free to ask me to clarify.
Yeah. So I'm gonna throw the diagram up in a second, but some of the words you just used just jogged my memory: odds ratios, and what was the other one? Because in my current government job, I'll just say that I work in pesticides. I won't go into much more detail, but I was a part of a science advisory panel a couple of years ago for a specific chemical, and part of our team involved epidemiology. And when my colleague got up and started talking about odds ratios and meta-analyses, my mind just kind of went, like, I don't know what she's talking about, but I was able to. I'm going to ask you about meta-analyses before we get done, but the more I listened to my colleague's part of the talk, the more I started to understand what it was all about. Okay, we have another special comment here. Corrine Henry says, you have a brilliant family, Miss Anderson. And my mother says, I'm bursting with pride, virtual hug. And we have another relative here. Good evening, Anwar and Mara. This is a great topic. So interesting. Thank you, Auntie Tracy. All right. So I'm going to throw the diagram up, and then I'm going to ask you to just kind of break down what epidemiology is for just for everyone who doesn't know. So I was perusing Google to find nice visuals for this. I figured you might bring some of your own, but I found this chart. So you see epidemiology up there at the top. You see monitoring, responses to diseases, investigating emerging diseases, and then you have all those things underneath like morbidity rates, mortality rates, determining risk, recommended control measures, determining causes of outbreaks, and instituting control measures. But you just said that it's in large part statistics. So how does the statistics fit with all those boxes that we just saw?
Okay, so epidemiology in essence is the study of diseases, how they spread, and ultimately how to stop them. Within that, you can do many, many different things. Broadly, you can work in a lab, you can work in the field, or you can work on the computer. And as I was kind of saying before, you can use things as building blocks. So when I was getting my degree in the control of infectious diseases, a lot of people that I went to school with had a microbiology background. So if you take microbiology and you add epidemiology, you can do virology, you can do diagnostic testing, so testing whether or not people have the disease. You can also go into vaccine development. A few people that I went to school with were veterinarians, and if you take vet and epi, you get something called One Health, which is focuses a lot on diseases that transmit from animals to humans. So things like leptospirosis or rabies. You can also have a medical background like an MD and take that with epi, and then you can be a clinical epidemiologist. Or you can do what I did and do public health and then take that with epi, and that's more like population level health. But you can also just go in a completely different direction. So on the far left of that diagram you had there was surveillance, which is a big part of epidemiology. So I'll wait for you to pull that up. Cool. Oh, that says monitoring public health. So that's basically surveillance, which is, what diseases do we have? And the word for that is, say, endemic. So flu is endemic, means that it's in the US every year. We expect it, and we just need to monitor it. We need to see how many cases we have. The mortality rate is pretty self-explanatory. It's how many people die. Morbidity rate is a little bit more complex. So that's how many people are living with the disease. You'll have to forgive me. I keep talking about infectious diseases because that's my focus, but do it with, say, cancer, which is not an infectious disease. So morbidity would be how many people are living and suffering with conditions of the disease without actually dying. And then epi, is can be a little bit more emerging. So looking at specific diseases, outbreaks, epidemics, things that are fast paced, but it doesn't have to be. Determining the cause of outbreaks and then control measures. So control measures have to be long term, because if you give up on the control measures, then there's a good chance that there'll be a spike. So an epidemic is kind of the opposite of endemic, which again means that it's there all the time and you expect it. An epidemic means that there are more cases than you would expect for a given area in a time. So if there were 2 cases of polio in the United States this year, that would automatically be an epidemic because what we expect is 0. And then also, yes, investigate emerging and re-emerging diseases to keep those controlled, determining risk factors and recommended control measures. So similar. So that's the far right is kind of long-term epidemiology, which is a lot more of the stats.
Okay. All right. Let me look at my questions here.
Please ask questions. I might be rolling through a little bit.
Okay, and if you guys have questions in the chat, feel free to ask them as well, and we will. I'll try to get your questions in as well. Let's see. So that's the field. What are the major? Let me go. The major subsections of it. I know you might have mentioned this already, but let's just do that again. The major other like a few major subsections, or can it just go, or does it matter? Does it matter which degree you combine the epi training with?
I mean, it matters to an extent, as in maybe if you got a degree in like music and epi, it wouldn't mesh quite as well. But really within STEM, if you would like to mix something, you can. Even if you come from like a stats background, you could do a lot with infectious disease modeling, which is incredibly useful. And modeling disease is kind of based on the data we have, estimating how many cases and what cases will look like moving forward. And that's really helpful. And again, I mentioned about like clinical work, field work, and then just standard stats and public health surveillance, which is also a big part of EPI.
Okay. Good evening, Shonda. Thank you for coming through. Shonda is, I believe she's a nurse. So, Tamera, so I know of epidemiologists in the public sector because I'm in the federal government, but where else are there other places where epidemiologists are employed? probably at universities, right, in academia, but can you
So
talk
yeah,
about that?
you're right. A lot of government sector, there's obviously tons now with the pandemic. There's also loads of research jobs. So like I've applied for quite a few research assistant jobs. So research is definitely a big facet of it as well. And also you can, again, like I said, get other training. So if you're a nurse and an epidemiologist, then there are roles for that as well. So I guess it kind of depends mostly on what you choose to build it with and what you'd like to be. A big place also is hospitals, if you'd like to be an infectious disease specialist. So obviously people come to hospitals presenting with symptoms of infectious diseases, and somebody's got to diagnose it because that goes outside of the realm of what general practitioners are tasked with being able to diagnose. So there's loads of different places that you can work in, but most of them are hospitals, academia, government jobs, and other research positions.
Okay. All right. Well, let's zero in a little bit and talk about what aspect of it you were and are interested in, and then we'll go and talk about some of your research.
Yeah, so like I said, I focus on infectious disease epidemiology, and a communicable disease is one that you can transmit from one person to another person. So the flu is an infectious disease, COVID-19 is an infectious disease, but something like cancer would not, because you can't transmit that from one person to another. So that's where I focus on, and again, population level things. So I focus a lot with environmental epidemiology, which, how are humans interacting with the environment? How does that impact our susceptibility to disease, and how does that impact the environment, again, in that kind of feedback loop. So a few of the classes that I took in my master's degree were environmental epidemiology, poverty, population, and the environment, as well as like broader things with policy, populations, and power. And then we took a class called IDAC, which is Introduction to Disease Agents and Control, which is basically going over like a few hundred infectious diseases and kind of learning about how they transmit. But my focus is definitely population level and environmental epidemiology.
Okay, so since you mentioned environment, I have to ask this.
Okay.
In the epi world, has 5G come up?
So that is also under non-communicable, because that would.
Oh, that's right. That's right.
But definitely things like radiation. I've not heard 5G specifically, but that's not saying that it doesn't exist. It's saying it's not my specialty, so I wouldn't know as much.
Okay. All right, because that was when this whole thing started. There were lots of 5G.
In my professional opinion, do I think there's any legitimacy? Probably not, but I will reserve that's not my specialty.
Same here. Okay, so infectious diseases. So just are there specific infectious diseases that you were interested in, or like are there well, we know what's going on now, but I think you probably started your studies before this whole thing started. What infectious diseases were you interested in? Which ones are you interested in now?
Okay. So my main focus for wanting to do epidemiology and public health as well is health equity, meaning that everyone should have the same access to health support. And ideally, we should all be at roughly the same risk of infection for diseases. And unfortunately, that's not happening because of social factors, which means that certain people are socioeconomically disadvantaged, which puts them at risk for certain diseases. And that's my focus. So my focus is diseases that present themselves in ways that are inequitable. So for my undergraduate research, my project was how conservative political ideologies impact vaccine hesitancy for the HPV vaccine in minority ethnic populations within the United States. I know it's a mouthful. But yeah, it was about like, well, how do government structures and what is perpetuated by them, how does that impact people and how they view science and how they view vaccines and things of that nature. But the research that I'm doing now and that I did for my master's degree, it's ongoing, was the individual, household, and environmental risk factors for chikungunya seropositivity within 4 structurally underserved communities in Brazil. Chikungunya is a disease. It's an arboviral disease. And an arboviral disease is one that's transmitted by an arthropod, which is like ticks, but usually when we're talking about them, we're talking about mosquitoes. So chikungunya is transmitted by the Aedes aegypti mosquito, and it causes arthralgia, so a lot of joint pain, and the actual word comes from a Kimakonde word, which means like doubled over in pain. So it's very painful. But the big problem with chikungunya, because you're not going to die from it, really, the mortality rate is exceptionally low, but the prevalence has gone up tremendously over the past few years, which is a huge concern that the burden is going up, and that is because of climate change. So more temperate areas, the temperature rising means that more areas can inhabit mosquitoes, and mosquitoes can be nice and happy and warm and transmit more diseases. So we're going to see a lot more arboviral infections coming up in the next few years, which is why chikungunya is a pressing concern. And how I conducted this research, well, initially it wasn't going to be this. Initially, I was going to be doing like a spatial video geonarratives in health for leptospirosis, but the pandemic happened, and then I had to move to a secondary data analysis. So there was a survey that was conducted within the 4 communities that I used, and that was conducted 2 years ago, and that asked questions about where they live, where they work, what kind of house do you live on, and do you live on a hillside? What material is your house made out of? Do you walk outside without shoes on? So things that cover individual household and environmental risk factors. And then along with that, there was an IgG test, which is a diagnostic test for chikungunya. And I did a secondary data analysis of that. And the first thing I did was a spatial analysis of the data. And you can pull up any of the maps, and it doesn't matter.
Okay, yeah.
Just so I can explain the...
So I have education, elevation, income, LC, Q, anyone?
Yeah.
All right, I'll do education. So what exactly is chikungunya? Is that is it a microbe of some sort, or is it?
It's a viral disease.
So it's a virus.
Mm-hmm.
Okay. Because when you said it's transmitted through a mosquito, I automatically thought about malaria.
Yeah, so malaria is also a virus.
Oh, of course.
So it's in the same category as like chikungunya, dengue, Zika, West Nile, etc. So these are my study areas. And this is a Landsat map. So basically somebody took a drone over the area, and this is what the area looks like. And then the little dots here are education. So the lightest dots are the less education, and the darker blue that it gets is the more education that somebody got. So this is how I spatially looked at my variables, and I did a descriptive analysis of that, which basically means that I looked at the data, looked at the seropositivity, and the seropositivity just means like a positive IgG result, which means that somebody's been clinically diagnosed with this disease, and just kind of looked at where it looks like the most cases are situated, and if there's any pattern with that. And then I also did a land cover data.
You want me to switch to that one?
Yeah.
Okay. Now when you say IgG, you're referring to an antibody, right?
Correct.
That's the detection tool. Okay. And this is what we're looking at. This was in Brazil?
Correct. So these are 4 communities within the northwest of Brazil.
Okay. Let me switch. So I will go to elevation.
Sure. I said land cover, but that's fine too.
Land cover is the LC, right?
Yes. Yeah. Sorry.
Okay, I knew that. I'll go to that one.
So something important about these 4 communities here is that they're structurally underserved, and in fact, the proper name of these types of communities, like the land type, is called a subnormal conglomerate, which is not a term that I use because I thought it was a little bit harsh.
Let me blow that up.
And basically what that means is that there was a lot of development, a lot of people moving into the area all at one time. And the rich people bought up a lot of the good land, which meant that some people had to move to other areas where they had to build their own houses on places where, are not ideal, the house is on a really steep hill, etc. And also the government just doesn't really give these people any funding, as in like trash sometimes is even picked up from these areas, are just kind of forgotten about for no fault of their own.
Mm-hmm.
So this is a land cover classification. So we divided it into impervious surfaces. So that would be things like concrete, basically. Water, pretty self-explanatory. There are like rivers going through. Oh, you can't see it on this one very much. But the bottom left one there's a river running through where like the little dolphin bit is. It's not a dolphin, but it looks like it. Vegetation, and then exposed earth. And I did a buffer around each house to see what they live closest to and if that would have any impact on their disease risk. So these are all exposure variables. And elevation was another one. If you could pop to that one really quickly.
Yeah. Let me blow that up for everyone.
Okay, so here the darker areas is the lowest to sea level. So if you see on the bottom right one here, it's completely black almost, so very close to sea level. And that one had over double the seropositivity rate of all of the other places. And the reasoning for that is because the water just kind of pooled in and sat there, and the mosquitoes were basically just having a party. But this is what the elevation looked like. And again, another one of my exposure variables. And then another thing that I calculated was something called a normalized difference vegetation index, so NDVI. And what that is it's a way of measuring plant health. So I think that most people know that mosquitoes like nice, warm, wet environments, but they also kind of have to host, and they host in nice, leafy, marshy areas. And it was thought that plant health would have an impact on where mosquitoes would nest. So if you could switch to the NDVI map while I continue to explain that. So that's what I measured. So basically plants give off infrared light, and you can measure how much infrared light they're giving off through drone imaging to see how healthy the plant is. So that's what I measured here. Excellent. So for NDVI, a value, it goes from -1 to 1. A value of -1 would be water. A value of 0 would be like rocks or another impervious surface. And then the higher the number, the healthier the plant is. So you can see that it's mainly on the outskirts of where the study areas are, with the exception of the top left, where it's nice and green in the middle, and there's like a fountain there. So after I did that, I kind of gathered all of this data and exported it to a data analysis software called R, which is where I did the rest of my analysis. And what I did with that is, one thing that I did is I transformed my variables. And what I did is I conducted what's called a principal component analysis for a wealth index. And the reason why that's important is because I wanted to measure how much wealth each person had in comparison to the other people in these areas. I think sometimes when we think about wealth asset comparisons, we think just in terms of like rich and poor, and that's not necessarily true. So there's something called like relative poverty, which is worse than absolute poverty. And what that means is if I am living in a community where everyone is making, say, $3 a day, so that's decidedly impoverished, I would be better off in that, better off is a crude word, but understand what I mean, better off in that environment than if if I was living in a community where the average income was, say, $20 a day, and I still was making my $3 because I wouldn't be able to afford things there and things of that nature. So it was important for me to gather relative wealth within these areas. So what I did is I gathered what assets they had from the survey, and I just divided that into quintiles, so fifths. And divided them up that way. And then I conducted the rest of my analysis, which is nice and technical, and I won't bore you guys through that one. But essentially what I found was that things like streetlights, living close to sewers, streetlights was a protective factor, which means that it made you less likely to have chikungunya. But things like living near a sewer, being Black or mixed race, being a woman, all of those things were risk factors for chikungunya in these communities.
Okay, so let's go back. So this analysis that you did, because it sounds like you were looking at a whole bunch of different things, and you put them all together for your analysis. Is this a form of meta-analysis?
No, not really. So this is just kind of like a multidisciplinary analysis.
Yes.
So in terms of using the spatial data along with the survey data, so a meta-analysis would be basically multiple studies. So for those that are not within, science academia, that you can never do a study once. It has to be done millions and millions and millions is an exaggeration, but lots of times to be able to say that, like, yes, this is what it is. So a meta-analysis is kind of, it's often done in lit reviews, but comparing results of other studies to make sure that you're getting the same result. So because this was only one study, it couldn't be a meta-analysis, but there was another similar study done in Pau de Lima, which is an area near to where these communities are, which I'm not gonna, the reason that they're blacked out is because this is still in the process of being published. So I don't know if I'm allowed to give you that information.
Yeah,
which is, so Pau de Lima is similar, is in a close location and like similar type of environment. But that still wouldn't be a meta-analysis. It's just kind of a comparison between those areas. But chikungunya was only introduced in Brazil in 2014. And unfortunately, the burden has been quite extravagant, which is why there are so few studies and which is why this study was so important.
Okay, so I'm gonna pull us back out to 30,000 feet for the audience. So chikungunya, that's a virus.
Correct.
Probably naturally occurring, not man-made.
Correct.
And so you went over. You mentioned a lot to us, but
I know. I'm sorry.
Your overarching question is what makes certain populations more susceptible to getting infected by the chikungunya? Is that what you were?
That's close. So what I'm looking at is what risk factors would make you personally more at risk for chikungunya. So again, one of the variables was walking outside without shoes on. So if I found that walking outside without shoes on was a risk factor, then that's something that we can say. But it's not about population. That one is about, again, individual, household, and environmental. So individual factors, stuff you can change, no biggie. Household can be a little bit difficult because some of the things were like wall material and floor material and things like that, which are a little bit more difficult to change. But most of the risk factors that I found were environmental, which means that that is a structural issue and that the people within these communities and communities like them need structural support. So like I said before, a lot of times epidemiology does focus on the individual or on the household, which there's benefit to that, of course, but if most of the risk is coming from things outside of your control, then that's something that needs to be focused on as well.
Okay, let me ask you this. Another epi vocabulary word I remember from my colleague at my job was confounders. Mm-hmm. Did you have any confounders in your work? So meaning that, and correct me if I'm wrong, that means was there anything that made your data more complicated or hard to interpret? Is that what that means in this context?
That is a rough and ready way to describe it. Okay. I think the technical way would be a little bit more, I don't know if it's worthwhile explaining, but that's basically it. A confounder is, if you're trying to go from A to B, a confounder would be something in the middle that thinks it's on the way, but it's kind of not. So it's kind of like a trick almost. And you can control for confounders, and you should control for confounders as early as possible. So I did that at the variable selection phase, as well as you can control for the confounders at every stage in the analysis. They can still happen. The idea is to control for them wherever you can. So one thing that came out that's not technically a confounder is streetlights. So streetlights was a protective factor for chikungunya, means that if you had streetlights on your road, you were less likely to get chikungunya. Now that seems pretty innocuous, right? Like what does that matter? The reason why it does is because streetlights in this circumstance were a proxy for income. So a proxy means that an indicator of. So living in an area that has streetlights means that you have more money because you can afford to live in that area. So really, when we saw that streetlights were a protective factor, the interpretation of that is that if you can afford to live in a place that has streetlights, then you're less likely to contract chikungunya.
Okay. Let's see. Do I have any more? And you're working on publishing this work?
Mm-hmm.
Okay.
So I'm redoing my analysis now, and we're adding some more technical things to it, as well as doing some modeling work for other communities that are similar to them. So again, the modeling is guesstimating based on the data we have, what would be the prevalence of this disease in another place or another time.
Okay, so in terms of, and I think this might be my last question because we're at 51 minutes. The data that an epidemiologist collects and considers, it sounds like you considered an antibody test. It sounds like you did some surveys as well. And I'm looking at this data set right here. These maps and the normalized vegetation index. So you looked at the ability of the vegetation to grow at different elevations.
This was plant health.
Plant health. Okay. So you basically pulled together multiple sources of data.
Correct. Yeah.
Okay.
But again, you don't have to do that. I could have just used the survey data. I didn't have to use the environmental portion. And there have been other chikungunya studies that did not use seropositivity, so the IgG and the antibodies, and they just used symptoms as a proxy for whether or not somebody was seropositive or not. But in my study, my outcome variable was whether or not the person had chikungunya. So I did need a way to measure that as my outcome. And everything else, all of the different exposure variables through the maps and through the survey were my exposure variables. Okay.
So your outcome variable, that's the thing that basically tells you, whether in this instance, whether they were infected or not.
Correct. So that's what you're studying. So the outcome variable is what is the end result? What am I looking at? So here I was looking at what's going to get somebody positive or negative for chikungunya. So that's my outcome. That's my end game. And the exposure variables is what are the things that could possibly make somebody positive or negative.
Okay. Gotcha. All right. So for those of you who are watching this, if you have any questions, feel free to ask. I have one more. And after that one, then we'll wind it down. But if you have any there in the audience, please feel free to ask. Ken Sean says love it. So all right. So my final question, it sounds like you're gonna publish this. Which journals do epidemiologists, is there a journal of epidemiology? I'm sure that there are several that you all publish in.
There are a lot. I'm not sure which one we're going to go to publish this one in, but the Lancet is, of course, a big one. New England Journal of Medicine sometimes if there's a crossover with medicine. And then there are like a few specific like infectious disease journals as well.
Okay. All right. So I'm going to bring this down, and then let's see. My final question is, can you talk a little bit about your future directions and professional interests? You know, where do you see yourself, or what are you interested in doing going forward?
Yeah, so I'm definitely interested in doing more research. So right now I'm in the process of exploring job opportunities, whether I'm going to be like a pure epidemiologist or a research assistant, that kind of thing. And I plan on working for a little while, but in the next year or 2 years, I'm definitely intending on starting my PhD in environmental epidemiology or global health epidemiology.
Okay. Okay. So environmental health. So you'll be looking at, for example, well, changes in the environment and what that due to.
Yeah, so that is a huge topic of interest at the moment, because, like I said, arboviral diseases, so diseases transmitted by mosquitoes, are going to be on the rise and are already on the rise. So it's definitely a big topic at the minute to look at the environment, how it's changing, and how that's going to affect our susceptibility to disease.
Okay. And I imagine that fits under the global
Global health, yeah.
health as well.
Global health projects are typically done in like low and middle income countries. So like I was supposed to go to Brazil, maybe I'll go to Brazil, but things like that, yeah.
Okay. All right. Well, Tamera, that's all I've got. I will probably have to listen to this a couple times.
So
Get everything.
no, please ask questions. I know I kind of railed through things.
Yeah. If people want to ask you follow-up questions, are you on social media, or do you? I mean, would you be open to answering any follow-up questions?
I'm happy to answer follow-up questions. I do not have any social media, so if they want to ask you, I'm happy to relay any answers.
Okay. So if you're watching this and you have questions for Tamera, then I'm the middleman, basically. So okay, that's fine. Well, Tamera, thank you for coming on and talking about epidemiology and your educational path and your research. I plan to have this channel going for a little while, so I definitely have to have you back if you're willing to come back and learn more about it. Okay. All right. Well, everyone, thank you for watching this discussion. If you're watching on the playback, please like, share, and subscribe. This channel right now is just over. It's at just over 800 subscribers, and once I get it to 1,000, once again, I'll have a super chat. But I think more importantly, I'll also have a community tab so I can share more information. So with that, I will shut this down. Again, if you have any more questions for Tamera, just reach out to me, and we'll go from there. So look for more content coming soon. And everyone, enjoy the rest of your night. And as always, remember that your attitude determines your altitude. Take care, and I will talk to you the next time. Bye-bye.
All right.
All right. Let's see.